Thank you very much for the nice introduction, and thank you for the invitation in this nice audience. And it's now an academic presentation after a couple of very nice company presentations to see how in academia, at least in my group and in Boston, in this particular project, we try to develop new immunotherapies for particular type of tumor. The project that I'm talking today deals around targeting one particular gene that is called ALK, that stands for anaplastic lymphoma kinase. This tyrosine kinase was cloned now 30 years ago, exactly in 1994, in one type of lymphoma. That's where the name stands for, which is called anaplastic large cell lymphoma. But then eventually was discovered in many other tumors, including non-small cell lung cancer, which is one of the largest type of tumor driven by this oncogene, and you see there is a long list. So ALK can be oncogenic via fusion, so chromosomal translocation leading to chimeric protein that basically have in common all the same portion of the kinase domain, and they are fused to different partners, and these are basically this type of tumors. But ALK can also be oncogenic being a receptor, which is basically normal function, right? And that's what happens, for example, in neuroblastoma, where ALK can be amplified or mutated or even just overexpressed by neuroblastoma cells and also by other tumors. For example, glioblastoma can be expressed, very rarely can be translocated, and other type of tumors as well. As a pathologist, we recognize an ALK-positive tumor by simple immunohistochemistry, because I will touch this point later, but basically normal tissue basically do not express, except with few exceptions. The ALK protein, ALK, is a receptor that is expressed during embryonic life, helps development of neurons, motor neurons, and enteric neurons, and of course is reactivated after this chromosomal translocation. So for example, here you can easily detect an ALK-positive lung cancer by simple immunohistochemistry compared to an ALK-negative lung cancer, and of course you see that all the normal surrounding cells are negative. We are particularly affectioned to this oncogene because we thought that there were several characteristics that made us thought that this could be some sort of an ideal target for immunotherapies, right? First of all, as I said, it is an oncogenic driver, so it's not just a passenger, and everybody of us know that many of the cellular immunotherapy in use today, including the very successful CD19 CAR and other type of CAR, they do not target oncogenic driver, they just target molecules that are expressed by tumor cells. And so one way for tumors to escape, of course, the immunological pressure is to stop expressing the target. In the CD19, for example, cells become CD19 negative, right? Relatively easy, I would say. In the case of ALK, we think that this is not possible because this is the oncogenic driver, so the tumor is dependent on the expression of this driver. All the cells, 100% of the cells express this target, so it's not subclonal or just partially expressed, but fully expressed by 100%. All the cells are strongly addicted to the neoplastic activity, and that's also confirmed by the fact that now there are 5 ALK tyrosine kinase inhibitor approved by the FDA, and there is one sixth coming. And importantly, as I said before, normal tissue, I will show some image, basically do not express ALK, and there is a very high selectivity expression by the tumors. One key point is that exactly because all these statements tend to be true, when patients develop an ALK-positive tumor, they spontaneously develop immune responses against ALK. They develop antibody against ALK, they develop CD4 and CD8 T cells specific for ALK. So there is, say, a natural proof that the immune system can be engaged to have an ALK-specific response. And so basically we develop a portfolio of immunotherapy because we know that targeting this oncogene as to create a portfolio. Some tumors, they express ALK in the cytoplasm, like diffusion. So lung cancer, for example, ALK is not expressed on the surface, but it is expressed inside, internal. In other tumors, like neuroblastoma, ALK is expressed as a receptor, so it is on the surface of the cells, right? So we need a portfolio of immunotherapy, and so through the years, we developed CAR T against ALK to target tumors that express ALK as a receptor on the surface. We develop a vaccine that basically targets tumors that express ALK in the cytoplasm, and peptides are processed, presented by class 1 and engaged T cells. Very recently, now we are also cloning and developing TCR T cells to basically try to have multiple tools to target this type of tumor, ready for potential escape or different mechanisms. Today I'm talking on this part on CAR T cells for a recent publication that explain what we developed. So what is neuroblastoma? Neuroblastoma is the most common extracranial solid tumor in the childhood. It's about 50% of pediatric cancer deaths, so it's probably the #1 cause of death in pediatrics. Low risk, they are quite favorable, but however, there are 50% with high risk, with a kind of dismal outcome. The classical standard of care, chemotherapy, eventual bone marrow transplant, and consolidation therapy. There are some antibodies, some type of immunotherapy with anti-GD2 antibody that can be used in this case. But overall, there is a large subset of patients that unfortunately do not respond. And so through the years, of course, there have been different type of therapies. I think many of you are familiar, for example, with GD2, that is a good target for neuroblastoma. There are CAR T cells against GD2. There are antibodies against GD2. The antibodies are approved, CAR T not yet. You can use radionucleotide type of therapy. And of course, you can go after ALK, because, as I said, ALK is an oncogenic driver for neuroblastoma. And so now in phase 1 trial, some ALK inhibitor, they start to show a good efficacy in these patients. And in neuroblastoma, there are different genetic flavors. About 10% of neuroblastoma have activating point mutation that make this receptor constitutively active. About 5% they have amplification of the receptor, and again, constitutively active. A majority of neuroblastoma, basically all neuroblastoma, express ALK in a way or the other. They have a wild-type receptor that, of course, can be activated by ligands and by other means. And once again, in other type of tumor like lung cancer and lymphoma, it is a fusion internal, right? So not expressed on the surface. So expression of ALK is pretty robust in neuroblastoma. You see this is mRNA expression, but also other pediatric tumor like Ewing sarcoma, alveolar rhabdomyosarcoma can express ALK to a detectable level. You can demonstrate by flow cytometry. You can nicely show by immunohistochemistry. You see that the expression of ALK is quite strong and homogeneous. Some rare cases, there is more heterogeneity of expression. And as a matter of fact, there is obviously a range, some very strong expressor, some lower expressor, but basically all of them are positive. Here, for example, is a comparison of normal ganglion cell, which are the putative cell of origin of the neuroblastoma cells. So you see that normal ganglia cells are completely negative for ALK expression, and in contrast, the tumor is nicely positive, right? So there is a nice gap of expression. In normal tumors, we screen a lot of normal tumors by, of course, available public data of RNA sequence, single-cell RNA sequencing, immunohistochemistry, etc. So you can look at the different expression. This is an ALK-positive lung cancer. This is an ALK-positive neuroblastoma. And you see that basically expression by RNA is extremely low everywhere. By immunohistochemistry, you don't detect anything except this is background. This is just colloid and debris. I'm a pathologist, so trust me. And the real expression that we were able to find is only on these cells. These are enteric neurons, right? So these are the cells that basically drive peristalsis and contraction of the intestine. And so these are probably the only real cells that really have a real positive for ALK. But everywhere else in the body, there is no detectable expression by RNA, by immunohistochemistry. So I basically already commented on all these points. A key point is that ALK kinase inhibitor are now in phase 1 trial. They show activity. A recent publication by the group of CHOP in Philadelphia showed that in particular third-generation ALK inhibitor called lorlatinib has a nice anti-tumor activity. So now we have an additional tool because this ALK inhibitor, of course, can be used to slow down tumor progression and delay like the growth of tumor, helping the introduction of any type of immunotherapy. But I'm going to show that lorlatinib has an additional unexpected property that we discovered recently in this publication. So we screen a series of anti-ALK antibody with different properties. ALK has a peculiar feature by which there is a cleavage site here. So ALK in normal cells is expressed as a long receptor or a basically cleaved receptor, and so we search for a CAR T that was binding to both portions to be sure to cover the entire spectrum of ALK expression in neuroblastoma and other ALK-positive tumor. And so our comparator were GD2.CAR, because GD2.CAR are so far the most active type of CAR T cells against neuroblastoma. And so here, for example, in one model in which ALK is nicely expressed, is basically a genetically amplified model. This is a human graft in NSG mice, metastatic. You see here how the tumor grows quite rapidly and metastasized to several organs, including the liver mainly. And one single CAR T injection was basically able to completely cure all the mice. We did histology in any tissue. The advantage is that by this anti-ALK immunohistochemistry that I showed before, we can detect basically even single residual cells in any tissue that are ALK positive, so that are tumor cells. And basically there was no tumor left, so mice were considered completely cured. So all good when ALK is expressed high level, but as any CAR, we challenge the system with models in which ALK is expressed very low, right? I show you some image of high and low expression. So when we go to the low side, neuroblastoma with lower expression, of course, now CAR T start to lose potency, but as well also GD2.CAR also don't really work in low expression. So we know that there is also a variety of response to the GD2.CAR. So you see that is a minimal response to low expression. And that's where the lorlatinib comes into place, right? So lorlatinib, at the beginning, we were combining CAR T cells with lorlatinib with the idea that blocking tumor proliferation, because that's what lorlatinib does, stops the activation of this RTK, and so basically stop an oncogenic activation and blocks the cells in cell cycle, and in many tumors induce a drug-induced regression of the tumor, right? So the original combination was just to combine two therapies that were slowing down the tumor. But turned out that we discovered, actually was anticipated by other papers, turned out that lorlatinib has an additional property that in some neuroblastoma, in particular the mutated cases, can upregulate the expression of the protein cells. So if you block the activity of ALK, there is a feedback loop by which the promoter gets activated, and so much more protein is expressed on the surface, right? And it is true in vitro, and it is true also in vivo. For example, this is exactly the same tumor by immunohistochemistry, which is a semi-quantitative assay, but still provides a clue. You see without lorlatinib, with lorlatinib, you see how the intensity of the staining. Of course, we validate this by flow cytometry, by multiple other means, and that's true. So what lorlatinib does, not only stops the oncogenic activity of ALK, but also stabilize and increase the production of the protein on the surface. So there is much more ALK available on the surface to target for CAR T cells activity, right? So we thought that this could be a very nice double effector with lorlatinib blocking the tumor proliferation, but also upregulating ALK expression on the surface and thereby facilitating CAR T targeting. And that, of course, all up to be true in models. We could clearly extend survival by combining lorlatinib and CAR T, and you see the same effect is not seen with GD2. This is a specific ALK-related effect, so it's not just blocking tumor proliferation and then adding a CAR T, right? No, it's really an ALK-specific effect on the surface of the cells. This was a cellular model, this was a PDX model, so we kind of nicely extend the survival. We could not reach a cure, unfortunately, because still residual cells survive, and that's the next level of developing even more powerful CAR in the next version that I'm not going to talk today, but that's one of the plan in the lab, of course. So based on these results, we are implementing a phase 1/2 trial in Boston, and this involves, of course, the clinical team at the Dana-Farber and Boston Children's Hospital, in particular Susan Baumeister, Susan Schusterman. We submitted this IND protocol to the FDA. I am the IND holder and the sponsor. It was approved in June, and basically we plan to start the trial October, November this year, so basically in 1 month or so, right? And the trial will be first starting, this is a first in human anti-ALK therapy, basically. Anti-ALK immunotherapy, of course, the ALK inhibitor already are approved, but is the first in human anti-ALK immunotherapy. And we will start, that's what the FDA requested, with very low dose and then increasing dose of ALK.CAR T alone first. And then if we don't see any particular toxicity, hopefully we won't, because in mice, I forgot to mention that one advantage that we had with this CAR construct is that this single chain fragment recognize both the human and the mouse ALK. So we could do a lot of I think meaningful studies for toxicity and persistence of CAR T cells in mice, because this CAR cross-react with the mouse receptor, right? And so the FDA was satisfied by those studies and considered enough for a potential investigation of toxicity. So, but still recommended to go with CAR T alone, and then eventually add lorlatinib to the maximum dose if no toxic effects are seen. And so the real trial for us will be the combo, right? The combo of CAR T plus lorlatinib for the reason that I just mentioned. So of course we will have primary objective, identify the maximum tolerated dose, and then safety and tolerability, and then the phase 2, which will be a sort of expansion phase, first CAR, and then in combination with lorlatinib, and then start to evaluate a complete and partial remission, of course, according to this criteria that are from the International Neuroblastoma Group, and then eventually do a series of correlative studies to understand the biology, the expansion, the persistence, and the potential toxicity of ALK.CAR T cells. So patients enrolled will be older than 1 year and below 30 years. We need, of course, confirmation of the diagnosis. We do not need a confirmation of ALK expression, because in our series, in other series, ALK basically expressed in more than 95 of the patients. There are few cases with very low expression of heterogeneity, but majority is quite good. I mean, intermediate to high expression and very, very uniform. So we are not asking an ALK staining for an eligibility criteria. And then, of course, we will study a series of characteristics. The trial will be like a standard one, screening, consent, and then we will manufacture the CAR T. The CAR T will be manufactured in the Dana-Farber cell manufacturing cell therapy facility. And that's where we did all the validation run for the FDA with donors. We did 4 validation runs. They were very consistent. The virus was produced in an academic environment, was produced at the Baylor College by Malcolm Brenner Group. And so it's a fully academic development of the product, right? And of course, the patient will be enrolled at the Dana-Farber and Boston Children's Hospital as well. And so we will manufacture the CAR T. We will have bridging chemo, of course. We will then do a lymphodepletion regimen as usual. We will start, when we will reach that point, hopefully, to combine with lorlatinib. We will pre-treat the patient with lorlatinib, because it takes a couple of days to upregulate lorlatinib of the surface of the CAR T cells. So we will basically start treatment with lorlatinib 2 days before the CAR T infusion, and eventually do the lorlatinib treatment either continuously, if not toxicity are observed, or basically by withdrawal and interaction by cycle of administration, with the idea of upregulating periodically the expression of ALK on the surface of the cell. And of course, there will be a monitoring program. That's it. So I think these are the content, and of course, This was developed, as I mentioned, in Boston in particular, now also in Torino. Monica, which is a PhD student, supported by Miltenyi. So thank you very much, Miltenyi, for supporting our PhD student that she's here. She's smiling there. So thank you very much. We hope she will be a next generation scientist in the Miltenyi environment. And of course, the group of the pediatric oncology at the Boston Children, Dana-Farber. And then a lot of collaboration. This is the cell manufacturing at the Dana-Farber. And of course, a lot of additional group that collaborated to bring this to the clinic. Thank you very much.
Thank you. Any question from the audience? One? Please go to the microphone so everybody can hear.
Thank you. Very nice presentation. I have a question to your mouse experiment. I guess you used CAR T cells from healthy PhD donors, right? Did you take in consideration that if you produce, you said you were looking for more potent CAR T cells, but now you use patient material, which will be considerably less potent?
Yeah. Yeah, that's of course is an issue with any trial when you move from healthy donor to patients. What might be reassuring is that neuroblastoma is one of the few tumors in which CAR T really seem to have some activity, right? So there are very nice data for GD2.CAR that has shown that neuroblastoma is a type of tumor, maybe because patients are young or they are different with certain type of chemotherapy, maybe because the microenvironment of neuroblastoma is a little bit more permissive than other type of microenvironment. Neuroblastoma cells are like a small round blue cell, and so they look like lymphocytes. So maybe that's one of the reasons possible. So the GD2.CAR trial are showing us that the product is not so bad in this even heavily pretreated patient, right? So we hope that we will get a product from this patient that will be sufficiently good.
Thank you.
Yeah. Short question, Fabian Müller from Erlangen. Thank you very much for this great talk and this overview. Is there any data, do you know of anything that the ALK inhibitor would directly or indirectly by modulating the tumor or stromal cells or whatever influence also the microenvironment? And would you expect something to happen by treating with both the CAR and the ALK together in that regard? Yeah,
that's a very good question. So in neuroblastoma, there is very little about that. There are much more data in lung cancer. So ALK-positive lung cancer, when it's treated with ALK inhibitor, clearly changes the microenvironment, right? And there are a few very simple reasons. One of them, for example, is that the ALK oncogenic activity drives the upregulation of PD-L1. That's very clear, both in lung cancer and ALK-positive lymphoma, right? So when you block by an ALK inhibitor the ALK activity, PD-L1 goes dramatically down, right? And so that starts to reshape the microenvironment because you kind of, as a side effect, you have some sort of activity against this inhibitory receptor, right? But then apparently there are very few studies actually, and almost nothing in neuroblastoma. Apparently inhibiting ALK also changing other factors in the microenvironment. The oxidative and the vascularization is changed. For example, ALK is responsible for the production of EFs, hypoxia factors, both in lung cancer and in lymphoma. And so when ALK is blocked, this hypoxia factor decrease, and so there is also a reshape of the vascularity. So there is an activity, still poorly characterized, but it's very possible that an ALK inhibitor will maybe help facilitating the activity of the CAR T cells by more tumor penetration.
Thanks, Roberto. Excellent talk. I guess the obvious question with the whole field and sector moving towards combination therapy, of course, is combinations. And so I wondered whether you'd looked at that in certainly your preclinical. We all know that monotherapies are not really going to work for solid tumours, but have you combined the ALK and the GD2.CAR T cells in your preclinical PDX models?
Yeah, that's what we are doing now. We already developed dual ALK and GD2.CARs. And I must say that so far they work great because what we observe is that the expression of GD2 is very heterogeneous in neuroblastoma. You have, again, very strong expressor tumors, and then like a smear of tumor up to quite negative neuroblastoma cases, right? And ALK is also is more homogeneous, right? And however can be lower level, right? So the idea of combining the two makes sense because you can have ALK that is specific on tumor. GD2 we know is when works very well. However, tumor cells can escape, becoming GD2 negative. So we already have them in different variety, and so far they work quite well. And that's one of the development that we want to do in the next steps. Yeah. Another development, of course, is similar to what was shown for the BioNTech with a different concept. We develop also a vaccine for ALK that eventually will be tested in ALK-positive lung cancer patient in a trial in the future. And that also can be combined with CAR T eventually, right? So that's another idea that we have in mind.
Hi, this is Remus from Miltenyi. Just adding on to the previous question from my colleague in Erlangen. Bryostatin also increases target cell numbers, especially for ALL. Nurali Shah has a CD22 trial and paper where using bryostatin increases the CD22 copy number. So there's we also show that it also increases the number of NK-like receptors on T cells. So that would be an example where a single agent modulates both the tumor as well as the immune cells themselves. Have you tried any other, perhaps, histone modification agents or other epigenetic factors? Because I think trying to box in the tumor with epigenetic factors might be a great way to inhibit them from drifting away from your therapy.
Yeah, we are doing that. The problem is that the promoter of ALK is still poorly characterized, right? So it is unclear, actually, I would say unknown, what are the factors that bind to the ALK promoter to upregulate the expression, right? Definitely there is this loop, so it must be something that depends on the ALK activity, etc. We tried some compounds, including EZH2 inhibitors, because in neuroblastoma they do a lot of things, including upregulating GD2, right? They do nothing on ALK, EZH2 inhibitor, they have no activity. So we are trying to test more compounds, but rather than going to compound, we want first to characterize the binding factor to the promoter is one of the line of research that we are doing now. So once we understand how the ALK promoter works, then we could find ways to upregulate it, right? But lorlatinib works not only by, it does upregulate the transcript, so definitely works in part on the promoter. But we think that the majority of the activity of lorlatinib is by preventing internalization, because many of these RTKs, in particular those that are constitutively active, tend to be internalized after activity, either ligand-dependent or ligand-independent, pretty much, right? When you deactivate by an inhibitor, you kind of block this internalization, and so basically ALK stays much longer on the surface, right, and accumulates there. So we think it's both transcriptional, but also by increasing the stability on the surface.
One question regarding the two approaches, the vaccination and the TCR T cells. They're both targeting the intracellular ALK. What is the advantage of one versus the other? What's the rationale between choosing between them?
Yeah, that's a very good question. Well, we know vaccine, when they work, they are great, but they typically in patients, they have like a heterogeneous level of response. Like some patients respond very well, and I think people from BioNTech know, and some patients tend to respond poorly, in particular those patients that maybe do not have an immune system that is ideal. Although the advantage of the availability of ALK inhibitors is that in lung cancer, and is moving also to the other type of ALK-positive tumor, now they are using first line. So basically these patients don't see chemotherapy at all. They are treated with, in particular ALK-positive lung cancer patients, they are treated with ALK inhibitor alone. So the immune system is in a very good shape, right? So probably the vaccination will be good. But the problem is that ALK is an endogenous protein, so there could be some deletion, by negative selection in the thymus, or basically selection of the T cell receptor with high affinity will be negatively selected, and so maybe the repertoire of the patients that express an ALK tumor could be skewed toward T-cell receptor with lower affinity, right? So when you vaccinate this patient, maybe you don't get a great response. If you clone an artificial TCR, and we already did it, that recognize with very, very high affinity, you basically bypass this problem in patients.
What's the chance that that mutates? Because if you have vaccination, you can have more, more chance to avoid escaping. But
yeah, I mean, we are not really worried about mutation, but rather by downregulation of class 1 molecule. ALK-positive lung cancer, they all express very high class 1 molecules, so that's not a problem. In contrast to neuroblastoma, in which class 1 is low, like many pediatric tumors, ALK-positive lung cancer class 1 is very high. But still, there is a chance, and actually maybe that's what will happen to like select a population that is class 1 low.
oh, one more question.
Very short question. I think you mentioned the ALK is also cleaved from the cell surface. Have you also tried to target this, maybe, by inhibitors? I think maybe it's
So
like...
to block the cleavage?
Yes.
Oh, we do not, because we think that actually the CAR T benefit from this cleavage. Because we have different type of cells that have the two receptor in different proportion. So some cell have a lot of the uncleaved receptor, and some other cell line have more of the cleaved one. And typically CAR T works better in cells that express the cleaved receptor. Actually, we are trying to do the opposite. We are trying to enhance the cleavage by introducing in the CAR T the enzyme, because the enzyme is known, the enzyme that does this cleavage. So introducing this enzyme in the CAR T cells so that when they reach the tumor, they also increase the cleavage and maybe they bind better.